Our last unit for our genome chapter, Unit 3, is eukaryotic genomes. So we've already given you a little bit of a sneak peek when we talked about C-value paradox and G-value paradox. But let's just review quickly. So eukaryotic genomes are very different from prokaryotic genomes and viral genomes by a couple of things. Number one, we have both nuclear genomes, which is the vast majority of our DNA, and then we also have organellar genomes. So from all eukaryotic organisms, this includes the mitochondria, which has its own genome, and for many other organisms there are additional organelles, such as the chloroplast in plants, which also has its own genome. And then for small groups there are even additional organelles that have their own genome. And of course the reason they have their own genome is because they originated as a symbiont, a bacteria living inside of a primitive eukaryote, and eventually became a portion of the eukaryotic organism that it can't survive without. And we've already mentioned number 2 here. Most of the eukaryotic genome is non-coding. This graph is the human genome, but it's roughly comparable for most eukaryotes, sometimes a lot more or a little bit less, but certainly the vast majority of almost all eukaryotic genomes is non-coding DNA. So in the human genome, only 1.5%, less than 2% of your genome actually codes for a transcription product. And most of those transcription products are then turned into proteins. Of the remaining 98.5%, about 25% is introns, non-coding DNA within the genes. About 50%, a little less, is transposable elements. So this green area, the orange signs, and the blue area, the lines, those are all different types of transposable elements. And then we have various other structural and other identifiable regions of non-coding DNA. So vast majority is non-coding DNA. And so a good portion of this comes from these transposable elements. Conservative transposable elements are important because they cause mutations and rearrangements, but they don't enlarge genomes. So we just simply chop out a transposable element and they code for the proteins that recognize themselves and cut themselves out and then reinsert themselves in other places. But non-conservative transposons or transposable elements are the most important. Sometimes these are called jumping genes. If this is like a cut and paste, this one's more like a copy and paste where we end up with additional material. So in addition to causing a mutation at the site you inserted, we also enlarge the genome. And so this is one of the main reasons. So notice in the human genome, roughly 50% of our genome can be assigned to one of these transposable elements. So that's a massive impact on the size of eukaryotic genomes. Now, there's been some question as to why do we have transposons? And that can be a tricky question to answer from an evolutionary point of view. There are some theories that it provides an advantage. But the question is, an advantage where? So as long as it does not provide a massive disadvantage for the organism carrying those transposons, the very fact that a transposable element can make extra copies of itself is an advantage to the transposable element. So it's going to be represented more and more and more as long as it's not too much of a drag on the organism that carries them. So they do also create new types of recombination, but probably playing the odds, these most often are bad, right? A random mutation, if you disrupt a gene, it's most likely going to be a bad thing. They do increase the chance of ectopic recombination, which can increase and make new combinations, but again, probably the vast majority of those are bad combinations. So there's some debate still as to whether there's an advantage for eukaryotic organisms for having transposons. The real answer might be that they're just these selfish genetic elements that are good at copying themselves, so they get overrepresented, and as long as they're not too much of a detriment to the organisms that carry them, then they are going to be kept around, and that's why we see them. So again, there's still some debate about that. There still might be some work and some research that might provide more information about whether they're an advantage for eukaryotic organisms or they're just an advantage to themselves. But again, they do occasionally create some recombination, some diversity and changes. And so just like in the prokaryotes, we can look at the synteny of eukaryotic organisms. So here are two closely related species of Arabidopsis. One has a smaller genome than the other, so that's why there's this jump up here. And then it's not quite a one-to-one ratio. There have been inversions and other losses and a couple of genes that have been moved around. But again, these are very, very closely related organisms. And so as we have examined genomes, we've known that not only for prokaryotes, but also within the eukaryotes, that genetic recombination, shuffling around of genes, movement of genes from one place to another in the chromosome is very, very common. We can see examples of this far and wide. So among the primates, humans have 23 pairs of chromosomes, but all the other hominids, right, so the extant ones, so gorillas, chimpanzees, orangutans, they have 24 pairs of chromosomes. So somewhere in the recent human history, we had a chromosomal fusion event. We still have most of the same genes that those other organisms have, but they're rearranged in a different order. And if we go a little bit more distantly, so this is looking at the synteny between corn and rice, which are both monocot plants, but have 100 million years or so of, maybe more, of divergence since they had a common ancestor. And so the large ones represent the chromosomes of one of them, and then color-coded and mapped to the chromosomal locations of where we find those portions of chromosome number 1, for instance, is found on chromosome 4, 3, 1, 8, 13, 11 in the other species. So massive rearrangements. So even though they might have 90% of their genes in common, there's very, very little conservation of synteny, right, of the order that these genes are found in. So that's a common thing that we see both in bacteria and in eukaryotes, that there's just frequent genome rearrangement. Now, eukaryotic genes, because they're so big, can't just replicate starting in one place. They have many, many places where they replicate. They also have structural elements like telomeres and centromeres that help to keep them organized during this replication process. And so this, and the fact that our genome is in different pieces, right, instead of in one big circular piece like in a bacterium, eukaryotes have chromosomes. So our genome is organized in linear pieces that are separate from one another. You can kind of think of it like volumes in an encyclopedia. So we've got many different volumes that all together make up our complete genetic instruction manual. We won't talk about meiotic drive here. That's not really critical for what we need to do. If you want to look it up, it's basically natural selection at the genetic level instead of at the organismal level, but it's not relevant for what we need to do. Telomeres are also structural. They're critical for maintaining the integrity of chromosomes and even play a role in aging. So as we get older, our telomeres get smaller and smaller, and that's one of the factors that causes us to do less well as we get older and older. Okay, so let's address introns, that other big chunk of non-coding DNA in eukaryotic genomes. Introns do have a very strong case for a selective advantage, and it's the fact that introns usually will break up genes based on their functional subunits. So what that means is if you look at a protein, there are different parts that we can identify. There might be a transmembrane domain and then a binding site, or there might be a enzymatic activity of one region of the protein and then a place where we bind a cofactor for the other part. And if we look at the introns, very often these introns are found aligned in between those functional units. So basically, proteins are kind of organized in the genome by functional parts. And what this means is that when a recombination event occurs, because the introns are there, it's very likely that these recombination events are going to mix and match and swap functional parts instead of just randomly swapping out one half of a protein for another. So for instance, if we had a bunch of functional parts from cars, like carburetors and engines and transmissions and all the parts that go into making a car, and they were taken apart by functional parts, so you had a complete carburetor, then if we put it onto another vehicle, even if it didn't fit exactly, there might be a chance that it would work and would be maybe not terrific, but might work okay. But if we had randomly just taken some sort of like a laser cutter and chopped up a vehicle into chunks without reference to the functional units, then there's almost no chance that one of those chunks would be anything useful if we tried to put it on another car. And so the same thing works for proteins. So introns have evolved to separate out the functional units of proteins. And so when unequal crossing over occurs, like we have down here, we get shuffling of different parts. We have these functional parts represented in the exons, separated by the introns that allow for mixing and matching and swapping of functional parts. And so although it's still not really, really likely, it's much more likely that we are going to have a functional recombination event when we have introns organized in this way. And so introns give eukaryotes an advantage and make more diversity and more combination when we do get recombination events. So this occurs for crossing over, unequal crossing over, and also for exon shuffling, which allows us, by other mutations, to move one part of a gene in functional units into another part of the gene. And like any mutation, these are mostly going to be negative and not provide an advantage for the organism, but we have a slightly better chance of them being advantageous. Now, as we look at GC content amongst the eukaryotes, we notice a different pattern than we see in the prokaryotes. So remember, in eukaryotes, we basically said that there's skewed GC content, where E. coli, for instance, might be 60% G's and C's, and another bacterium might only be 20% or 25% G's and C's. But those are fairly consistent across each organism's genome. Eukaryotic genomes are different. We do have GC content bias, but it's regional within our genome. So this graph illustrates that. There are areas with fairly low GC content, and there are areas with fairly high GC content. Overall, it balances out to roughly 50-50. But these unique areas are identifiable. This identifiable area is called an isochore. So an isochore is a region of a gene, or a region of a chromosome that is chemically different because it has more G's and C's, or some other chemical factor, but G's and C's is a very common way to define an isochore. So know what an isochore is, and know that GC content is biased in these areas. And so this shows that there's some sort of a selectional process acting on that region of the chromosome that's probably different than on this part. And so we have more intragenomic variation amongst eukaryotes, right? That means that G's and C's can go up and down across different regions. But there's not that much intergenomic variation. So if I compared a human to a cow to a fish, we'd see roughly 50 to 60% G's and C's across all of them, despite having these regional areas where it could be elevated or lower. And so this question here really, this question here really we're going to leave unanswered, because there's still lots of work that needs to be done. No one has a really firm, decided answer about what it is. If we were looking at one specific isochore, then we might say, okay, well, this is why that one's there. We might have specific information about that one. But if we looked across, it would be hard to say there are general rules that can explain all isochores. So just know that the selective forces that cause these different regions, these isochores, are varied and may be different from one isochore to another. Okay, that's it. That's all you need to know about genomes. You have your SLOs broken down into these individual subunits so you can study them and review them, go over them. If you have any questions, please feel free to reach out.